So, hello, dear guys. So, once again, I want to greet you in this lecture. So, today we will continue discussion in pathophysiology. This is topic known as general pathology of infectious diseases. So, uh, Infectious diseases are much more details explained in infectious diseases. You have a distinct subject about it. Now also you have microbiology where some details of this will be revealed. But now from the pathophysiology way, so you already know what is immunology, you know what is the inflammation and which reactions present in inflammation. So you have to understand general idea of this infectious disease. Okay, group number 5 finally decide to join. So let's talk about it. General principles of microbial pathogenesis. So infectious diseases are an important health problem in the United States and worldwide despite the availability of effective vaccines and antibiotics for many types of infections. So there most for United States very important influenza and pneumonia combined are the 8 leading cause of death in the United States. But exactly so in this book they said that infectious diseases are very dangerous and it's very common, yeah, and a lot of people die because of it. But main idea I want to share to you that actually nowadays infectious diseases are less common, so became much less common diseases which cause the death. So a few decades ago, so I mean before invention of antibiotics or safe antibiotics like penicillin, so this rate of death due to infectious diseases was much higher, and it was the first reason of death. So if we remember some diseases like cholera, yeah, cholera disease, so like Vibrio cholera was very widely present in our in countries, especially southern countries like Pakistan and India and Africa, descent people, and even in Europe we could find in Kyrgyzstan also very common disease, cholera, Vibrio cholera. But after invention of antibiotics or after invention of other methods of treatment of the diseases, we can most of people survived after these infections. Yeah, even if we have infections, so they are not life-threatening, so predominantly. But anyways, few infections are still very dangerous for us. They are here in this preview, it said that it's um, influenza bacteria, so also HIV, immune deficiency virus, yeah, human immune deficiency virus, and AIDS, yeah, so which cause also very widely present in all developing and also developed countries. So sorry. Diarrheal diseases like Escherichia coli, which were present even in European Union, very spread there. So a few years ago, so we had a pandemic of Escherichia coli bacteria, yeah, so which caused diarrheal diseases. Also, um, this still danger of infectious diseases are very common, and now, especially now, we have to discuss about it in viral infections. So when we can't treat viruses yet, and that's why we have this very tough trouble with the very tough struggle with the viruses we have, so like coronavirus. So we don't have effective treatment yet. And because reasons are different, might be political reasons because we don't pay, governments don't pay money for research of antiviral drugs. Also, it used to be that viral infections are less dangerous than microbacterial injury, and they cause less adverse effects, and usually our own immunity kills it easily. So that but anyway, right now we see, so in this year in 2020s, so that coronavirus really so really surprised us, and now we all of us are suffering because of it. Even because of it, we are in online education. Yeah, just because of this infectious disease which are spreading all over the world. So. And here we have to understand the principles of microbial pathogenesis. So let's talk about categories of infectious agents. So which categories of infectious agents you have to know? They are, so start with a less known and less very rarely present disease, but still they're present. They're prions. Prions. Prions, it's not classical infection. It's not a virus. It's not self-renewing cells, self-renewing DNA or RNA. It doesn't contain RNA or DNA. Prions are Brian, so there are proteins. There are proteins, so which change the conformation of other proteins in a cell and causing the chain-like reaction in the body. And some of these diseases are like Kuru disease, which is associated with a human cannibalism, still present in Papua New Guinea, so country, and also Creutzfeldt-Jakob disease. So we're also not very common, but it's a very common example of prion disease. Bovine spongiform encephalopathy. Also it's caused by mad cow disease, and variant Jacob disease. It's transmitted to patients, to humans through consumption of meat from BSE. So due to bovine spongiform encephalopathy or mad cow disease. So you see, mad cow disease is affecting cows usually, but when we eat them, so it causes Creutzfeldt-Jakob disease. So variant Creutzfeldt-Jakob disease in patients. Um, Usually, prion diseases found in neurons. So it main idea they cause conformation changes that confers resistance to proteases. So proteases can destroy it. And you remember that if misfolded protein appears in our brain, so we have auto degradation by proteasomes. Proteasomes, which destroy these proteins, misfolded protein, and then proteins are not spreading. But in these diseases like Creutzfeldt-Jakob disease or Kuru disease, these proteins are resistant, and they continue degradation of proteins in the body and causing the formation of more misfolded proteins in the cells. And so Creutzfeldt-Jakob disease can be transmitted from person to person iatrogenically by surgery, organ transplantation, or blood transfusion. Okay, yeah, and in details they are discussed in chapter 23, so we'll discuss it in next year, I guess, not next semester, but next year. Okay, let's skip it, but you have to understand that prions, they are not viruses, they are not some enemy. Yes, they're our own proteins, or might be proteins of other people, other person, so which can cause the infection, which leads to very severe brain damage. Okay, secondary, so let's talk about viruses. So viruses are not alive. So keep in mind that viruses are not alive. They are not cells. They don't have cells. They have only genetic information, which is surrounded by proteins. And this protein code it is known as capsid, and they can divide without help of cells. So they are spreading only inside cells, and that's why they named as obligate intracellular parasites. They totally depend on host cells' metabolic machinery for their replication. They don't have their own DNA transcriptase. They don't have um, their own ribosomes, and to produce protein of capsid and to produce RNA or DNA, so it depends on which type of virus is this. It uses their own uses host's own machinery, host's own yeah, a fabric of of synthesis. So because it can work. Okay, examples. Yeah. So example, poliovirus causing poliomyelitis. Yeah. Okay, let's discuss it later. There are numerous amounts viruses like cytomegalovirus, or might be just usual coronavirus. So not COVID-19, but other types of coronavirus are there. Adenoviruses. We have HIV. So you see, they affect absolutely different in different way. So there are a lot of different stamps of them, so which affect us in different way. But main feature of these viruses that they have their site of injury. They have their specific attachment areas by which viruses can enter into the cell. So we always have some protein on the surface of our cells, which can assist in the transmission of this virus into the body. That's why viral infections, they usually affect only one system. Usually affect one system. So as I said, yeah. So for example, so coronavirus, they usually affect only respiratory tract. HIV, it affects our immunity cells. Yeah, only CD8 positive T lymphocytes, no others. So and also and effects which generalized effects of viruses there happen due to our autoimmune reactions against it. So but usually viruses affect only one organ. So like hepatitis, yeah. So you know that hepatitis virus, it's present in blood, but it's not evolved in blood. It's just present in a way of sharing between hepatocytes inside. Okay. um, so and spreading pathways of viruses are very different. So there we'll discuss ways of spreading very soon. Then okay, general consideration of viruses we have. So it's very small compounds which sharing from living cell to another living cells, but their own are not living. Okay, let's talk about bacteria. So bacteria, they are prokaryotes. So usually when we say about microbes, they are usually bacteria, and these microbes, these bacteria, they are membranous. So unlike eukaryotic cells, they lack of nuclei. And some other membrane enclosed organelles like mitochondria. They don't have Golgi apparatus. They don't have so rough endoplasmic reticulum. Instead of it, they have free ribosomes. Instead of nucleus, we have a coil of DNA. Yeah, so that's the point. Okay, bacteria are classified by gram staining, so it's might be positive or negative, and according to this, we separate cells. So it's due to a specific structure of the cell membranes and cell wall. This is gram staining. By shape, we can divide them, and they might be spherical, which are known as cocci, or rod shaped like bacilli, and other shapes. You will discuss it in microbiology. And we can classify them by their requirement for oxygen. So most of bacteria, so which we encounter in our life, they are anaerobic. They don't, they can't survive with oxygen around. But some bacteria, they can survive, and also they can use oxygen in their life, and that create some special features of them. Okay, some bacteria are motile, some of them non-motile. So motile bacteria have flagella, long helical filaments extending from the cell surface that rotate and move the bacteria. Actually, this flagella is also protein, which creates additional antigen to this bacteria. Uh, so and some bacteria possess pili, another kind of surface projection that can attach bacteria to host cell or extracellular matrix. Bacteria synthesize their own DNA, RNA, and proteins, but they depend on the host for favorable growth conditions. If we talk about bacteria which cause diseases, but you know that there are a lot of bacteria which don't cause disease. Bacteria are present everywhere in our body, everywhere in our environment. We inhale bacteria every day. We eat bacteria, so with the food. So we just we encounter, so on the surface of our skin we have normal microflora bacteria. In digestive tract we have microflora bacteria which don't cause disease. They are normally present, and in soil we have, in water we have bacteria. So nothing is pure of bacteria. So this is, but this most of bacteria, they are not dangerous for us. They don't have in their cycle of work. This bacteria doesn't use human because they're self-sufficient. They can survive even without killing anyone. So, so 1 minute. Okay, and as well, they're self-sufficient. They don't have to kill us, and they don't work. So, and actually, desire of every. So, oh yeah, one thing I wanna confirm and wanna explain it to you. So that main role of main desire of bacteria and viruses is not killing us. They are not enemies for us. They are not. They are not just pure evil for us. Yeah, because they wanna kill us. No, they all of them are parasites of us. They're just parasites, and they just use us as a carrier of nutrients, and they use us for their own life. But point is that sometimes due to our own immunity response, or might be uncontrollable growth of this bacteria, so we die. So but it's not the main not a main feature. It's not the main desire of this bacteria to kill us. So they predominantly they want us as a carriers and spread, and they wanna use us as a spreader. So they like one who spread this their own genetic material all over the world. Okay, and now actually human body become so humanity is a very good tool for our bacteria and viruses or parasites to spread this bacteria in very fast way, because we have a lot of social binds, a lot of social bonds, and then every year, every few years, there will be a lot of new more infections. So be sure in it. So a lot of new infections will be there, and this bacteria and these infections will be more severe one after so every next time, because it's too good source for viruses to let it to so not to use it. So that I hope you understand. Okay, which so examples of viruses are there? So okay, and let's skip it. So bacteria. Uh, so yeah, and we have different types of bacteria. So let's talk about bacteria types. Many bacteria remain extracellular when they grow on the host. Sometimes, so most bacteria are like this, but others can survive and replicate both outside and inside of host cells. And they are known as facultative intracellular bacteria, such as mycobacteria, tuberculosis, and some grow only inside host cells, like obligate intracellular bacteria. So you see, these examples are rickettsia or chlamydia. So, and they are intracellular. So same as viruses, they use our own cells to kill to spread their genetic genome. Okay. Bacteria cause a range of infections, from common pharyngitis and urinary tract infection to rare diseases such as leprosy. Okay, here we see chlamydia and rickettsia are obligate intracellular bacteria that replicate inside membrane-bound vacuoles and epithelial and endothelial cells, respectively. These bacteria get most of all of their energy source ATP from the host cell. So you see, like viruses, they use our own metabolic mechanisms for their own survival. So chlamydia trachomatis, so as well I am urologist, I'll explain it to you. So they are a frequent infection in cause of female sterility by scarring and narrowing of the fallopian tubes, and blindness in case of chronic inflammation of conjunctiva, and eventually causes scarring and opacification of the cornea. But the point is that chlamydia carriers of chlamydia are males too. Of course, chlamydia trachomatis is very commonly present in males. It causes urethritis, very mild urethritis, and usually sometimes patients males they don't care about, so yeah, something itchy in urethra, so then they don't go to doctor to treat it, and then by sexual intercourse they spread this chlamydia to females, and in females it causes very sometimes it causes very severe effects, like as I said, female sterility, or maybe it causes miscarriages, so this chlamydia infection. And according to my experience, a lot of patients they deny this chlamydia. They claim that yeah, chlamydia is not dangerous because it's present in every second person, but actually it's present only in that persons who used to live their very open sexual life without contraceptives. So that in this case, and of course it's a very widespread disease, and it's very hard to treat it, because if we have bacteria which are not intracellular, we can clear it. We can just clear it. So with easy antibiotics, with easy antibiotical treatment, we can clear it. But in case of chlamydia and rickettsia, so they're intracellular. They're intracellular, and to kill them, we have to manage a lot of antibiotics, especially macrolide-like antibiotics, which can penetrate inside the cell through the cell walls, through the cell membrane. Okay, let's talk about rickettsia. So it's much more common in, if I'm not mistaken, rickettsia are not prominent in Kyrgyzstan and Asia. And so rickettsia are they're more commonly in Rocky Mountain. So that's why it's called Rocky Mountain spotted fever, and very common in the USA. It's rickettsia transmitted by arthropod vector, including lice, ticks, and mites. So this and all of them are causing rickettsia. And we also have mycoplasma and related genus ureaplasma are unique among extracellular bacterial pathogenesis in that they don't have a cell wall. They are tiniest free-living organisms known. So why we pay your attention that they don't have a cell wall? Because a lot of antibiotics we prescribe, they are like penicillin. They are mainly aligned to destroy cell walls. And these cell walls, destroying of cell walls, usually cause the death of bacteria. But in mycoplasma and ureaplasma, so they can survive without cell walls, and then cause the as it is not followed with death. So that makes it very challengeable to treat these infections. Fungi. Fungi are eukaryotes, so they're in the middle between plants and animals, which is more closer to animals. So they're eukaryotes with thick cell walls composed of complex carbohydrates such as beta-glucans, chitin, and monosyllated glycoproteins. So okay, let's talk about. Before fungi, I want to explain the structure of cell wall of bacteria. So this is a typical gram-negative cell wall, and this is a gram-positive cell wall. So here, as both of them, they have a cell membrane. Everywhere they have cell membrane. So in a gram-negative type, so they have 4 layers of cell membrane. So 4 layers of cell wall. They are inner membrane, peptidoglycan, then outer membrane, and lipopolysaccharide capsule. This and these lipopolysaccharides, you remember, yeah, we discussed in infectious inflammation. I said that lipopolysaccharides, LPSs, they are also known as they can cause fever. Yeah, they are pyrogenic agents, and also they can work as a chemokines to attract more attract our neutrophils, or so usually neutrophils, but in other leukocytes to the site of inflammation. Yeah, and these fractures of this lipopolysaccharides on the surface, or bacteria itself, it causes activation of our immunity cells. And this is typical appearance of gram-negative bacteria. And in gram-positive bacteria, so they also have cell membrane, but it contain only three layers: cell membrane, then peptidoglycan layer, so which is also antigenic for us, and also we have capsule, which is composed of glycoproteins. So here we see adherence proteins, teichoic acid, so which is also present here, it's mucopolysaccharide, and also we have M protein. It's an immune evasion protein known as M protein. So okay, in details you'll discuss it in immunology. Okay, let's talk about fungi. Question?
Yes, sir. Which sir bacteria will be more resistant against antibiotics, sir? Gram negative, gram positive, sir.
Answer is both. So it doesn't matter. So it depends on type of antibiotic and type of bacteria. So they are much more resistant. So for now, so it used to be said that gram negative are more they have
Resistant.
antibiotic treatment, but you take a look. So all these bacteria, they have very abundant ability for cell division, and every cell division is followed with gene mutations, and these gene mutations might be produced so during the evolutionary process of their cell division. So might produce some specific biochemical features which produce some enzyme which can destroy any other antibiotic. So it doesn't matter it's gram negative or gram positive. So that's why we can say so which of them are more resistant. Yeah, against penicillin. Yeah, of course, more typical gram positive. Some gram positive bacteria, they can survive. They can produce beta lactamase, so enzyme which destroy penicillin, or other remnants of all other compounds in a group of penicillin, because they have some special features. Okay, we'll discuss it I guess very soon. No, you'll discuss the microbiology actually, so it's much better. But
Sir,
many ideas,
sir,
but any of them so can be effective against against antibiotics. Yes.
sir, lipopolysaccharide is sir endotoxin, sir.
Mm-hmm.
Sir, if we use antibiotic against gram-negative bacteria and the gram-negative bacteria become destroyed, sir, in that case also lipopolysaccharide can cause the disease, sir, because of endotoxin.
And we will discuss it. Yes, that's true. So actually, we have specific types of effects. Actually, so when a lot of bacteria died, yeah. So when we treat patient, but in case of treating of patients, so a successful treatment of patients, his symptoms might become more severe even after treatment. It happens especially so specific symptom in syphilis. So when treatment goes well, so more cell walls are destroyed, and symptoms became more severe than without treatment. And that's why sometimes patients with syphilis, they deny the treatment, and they stop treatment because and they claim that it gets worse. Yeah, so they feel that it gets worse after treatment, and that's why they stop treatment, and then live with this sometimes live with this syphilis. But actually it is a symptom that things goes better. So yes, and actually when breaking of a capsule happens. Okay, first of all, Amjad, yeah, if I'm not mistaken. So if we have so we'll discuss what is endotoxin, what is exotoxin, and then I'll make this consideration. But yeah, you can keep it in mind. Fungi. Fungi, they're eukaryotes, as I said, they're in the middle between animals and plants. They have very specific structures. So they contain on the surface they contain carbohydrates like beta glucans, ketene, and monosylated glycoproteins, which are also very antigenic for us. Calcofluor white. So okay, let's talk about examples. Okay, some fluorescent stains that binds chitin provides a useful way to identify fungi in patient specimens. Um, so like here you see, so in the more angioinvasive mucor species, which are present here, so they're well colored here. Okay, um, okay, this structure is a fungi. Okay, so let's talk about which infections are caused by fungi. There might be superficial infections, so usually they're present in skin, hair, nails. They cause dermatophytes. So this it can cause tinea pedis, also known as athlete's foot. Tinea capitis is a scalp ringworm, so they can invade subcutaneous tissue, causing abscesses or granulomas. But usually, yeah, chronic infections, often in the foot, are called myxotomas. Myxotomas. So you see, they're usually superficial because our immunity works against it very well. So and they are restricted in areas where our immunity cells cannot reach. But sometimes we have deep fungal infections. They can spread systematically and invade tissues, destroying vital organs in immunocompromised hosts. So you see, immunocompromisation of host. So, for example, in HIV, the HIV virus, yeah, they can these superficial infections can delve deeper, and they can penetrate because of insufficiency of our immune system. But normally our immunity very well preserved against fungi. So fungi are divided into endemic and opportunistic species. So what is endemic fungi? Endemic fungi is they're limited to particular geographic regions. So, for example, in the United States in southwestern part of the United States is a coccidioides, and histoplasma in Ohio River Valley. So, which examples from our countries we can say? So I can't explain. I can say about it because there are no some specific mycobacterial infection, no specific fungal infection we have in our country. Might be in your country we have. I don't remember. And we have opportunistic fungi. Opportunistic fungi which are present in a lot of people, so only when they get immunity compromised. So the most obvious is Candida, Aspergillus, Mucor, or Cryptococcus. So Candida albicans, you know that Candida albicans, it's a very widespread disorder, especially in females, in case of vaginitis, so in inflammation of vagina. So it's caused by Candida albicans. So which is it is normally present in our on our body, so it maintains the integrity of skin a little bit. It causes some kind of protection for us. But when it appears in our mucous layers, in oral cavity or in vagina, which are similar through the histological review, yeah, this Candida albicans can spread and form in the some troubles for us, like itching there and erosions and some kind of lesions present in areas of mouth and vagina. So we have Aspergillus, Mucor, and Cryptococcus, which are usually present in AIDS patients. So if we have this, if we you can see that patient suffering with the Aspergillus, Mucor, or Cryptococcus pneumonia, you can suspect for AIDS. And the most typically for AIDS we have Pneumocystis jivareti. Giborecci, or previously it's called Pneumocystis carini. So this very specific infection for AIDS patient. Then let's talk about protozoa. So which kinds of protozoa you have to keep in mind? Protozoa, they're animals. Yeah, they're small animals. So might be single-celled eukaryotes, or might be yeah, a single-celled eukaryotes. They're animals according to classification of taxonomies. Yeah, they can replicate intracellularly, or might be extracellularly. So let's talk about them. Plasmodium. So, for example, Plasmodium, which causes malaria, yeah, they are spreading. They are dividing inside the red cells. Leishmania. They are spreading in macrophages. Leishmania is very common in southern Asia. Yeah, I don't know what is the epidemiological situation right now within Leishmania in India and Pakistan, but I knew that in 80s it was very widespread disease. And extracellularly in urogenital system, so, for example, it's a Trichomonas vaginalis. So it's another sexually transmitted disease. So it is parasite, often colonize vagina and male's urethra, and that cause very that cause urethritis and vaginitis also. So prevalent pathogenic intestinal protozoans are Entamoeba histolytica and Giardia lamblia. They're ingested as non-motile cysts in contaminated food or water and become motile trophozoites that attach to intestinal epithelial cells. And okay, we have bloodborne protozoa like Plasmodium, Trypanosoma, Leishmania. So, and will be discussed about them. Um, they replicate before being passed to new human hosts. And also don't forget about Toxoplasma gondii. Toxoplasma gondii, so it's mainly present, so actually very widespread disease and underestimated disease. So Toxoplasma, is very widespread in carriers of cats. So because a lot of cats are infected with it, and because Toxoplasma, it affects our brain. So Toxoplasma affects cat's brain and also affects mice's brain. First of all, it's necessary for it's present in cats because of what? Because mice with this Toxoplasma, they have a they can affect their brain to act as they don't afraid of cats, and they get revealed. Yes, as mice get revealed, and then cats are very easily can capture this mice, and they when they eat it, then all this all this they can be ingested to the cat, and in a cat it causes the formation of new cysts in the digestive tract of cats, and they spread it with the fecal masses. And you know that all carriers of cats, you understand me, so you just you petting them, yeah, so you do some you live along with them, so and then you also you can be affected with Toxoplasma, and in some patients Toxoplasma causes infertility in females especially. So Toxoplasma causes infertility, and that's very bad effects, and that's why be very careful with cats, and don't let them to eat mice, especially mice of I mean widespread mice, or if you carry the cat just for eradication of mice, yeah, so don't let them to eat close to you, don't let them to be to sleep in your bed, yeah, of this cat. But if you carry cat just for petting, yeah, so just for their snoring, yeah, so please keep them So keep them away from wild mice, because wild mice or rats, they can be carriers of toxoplasma, and then you won't be affected. Okay, and finally, let's talk about helminths. Helminths are parasitic worms. They are highly differentiated multicellular organisms. So these examples are Ascaris lumbricoides. So in immature stages, known as Toxocara canis, and Echinococcus. And Echinococcus, yeah. So what is so there we have to discuss. Okay, no, we won't discuss it. So it's very complicated. Roundworms. So helminths comprise three groups. There are roundworms, also known as nematodes. So, for example, lumbricoids, strongyloids, and hookworms. They are uh, they usually affect our digestive tract. Then tapeworms. So, for example, scolex, so they contain scolex and proglottids. So an example is Echinococcus. Of course, Echinococcus or might be Alveococcus. So very commonly present in Kyrgyzstan. So it's one of the one. Kyrgyzstan is endemic for this Echinococcus disease because lices, which present in our forests, they can trap this echinococcus, and they spread by fecal mass. They spread scolex, their heads, and and hunters through lices. So they can be infected with this, and this echinococcus usually after the digestive tract it spreads to the liver by portal vein system, and in liver it causes the formation of cysts, and these cysts they are growing and they can rupture and cause an anaphylactic crisis, anaphylactic reaction, and then the patient dies. And usually it happens. Okay, about echinococcus you have to read it by yourself. So very interesting disease in in very interesting in Kyrgyzstan. It's very commonly present. And also don't forget about cysticercia, cysticercosis. So this is very relative to echinococcus. So but it's spreading by porks. So by porks, yeah, and this cysticerci, they are present in um, uh, so you can find them in meat of porks, and when they enter in our body, so they can cause formation of two different forms. They can form worms, so like tapeworms in our digestive tract, or they can affect our muscles, and we have cysts in our muscles. Of course, it's also very strange disease, and we can treat. Yes, we can treat it very easily with few albendazole capsules. But anyway, if untreated, it can cause death. And finally, we have a flukes. They're leaf-shaped flatworms with prominent suckers, and that are used to attach to the host. So, for example, we have liver and lung flukes and schistosomes. Schistosomes are also typical examples of these flukes. Then let's talk about ectoparasites. Ectoparasites are like lice, bedbugs, fleas, arachnids, yeah. So like mites, ticks, spiders, and they cause disease by biting or attaching to and living on or in the skin. So they usually we name them as bloodsuckers, yeah. So there are a lot of bloodsuckers, and they are not dangerous for us without infections which they can carry. So, for example, um, so which example we have? So you know that arthropods also can serve as vectors for other pathogens such as Borrelia burgdorferi, the agent of Lyme disease. So which is very typical for USA. Yeah, any questions?
Mosquito, mosquito carry by.
So mosquito carry what?
Sir, plasmodium.
Yes, absolutely right. Some types, a specific type of mosquitoes, they carry causative agent of malaria. Yeah, and that's one of way of spreading of the disease. Okay. And these ectoparasites, they are not so dangerous itself, but they are more dangerous. For example, typhoid fever might be spread by ticks. Yeah. Um, so that's yeah, okay, that's enough. You understand that who are ectoparasites and what's the danger of them. Okay, and let's talk about microbiome. Microbiome, as I said, microbiome is normal bacteria which are present in our digestive tract or in the skin. They are helpers of us. They help us to survive properly. So they can digest food which we can digest. By concurrency, they are present on our skin, and this skin bacteria they prevent other bacteria entering our body. So by the formation of, for example, milk acid bacteria, lactic acid bacteria, so which can produce lactic acid on the surface of our skin, and they protect our skin from entrance of other bacteria inside us. So that's why microbiome is very important. So also microbiome of vagina we have, yeah, so which prevent same as we discussed and fungal infection like Candida albicans is prevented. It will prevented by bacteria if we have normal microflora. It is well protecting us against Candida albicans. But if you use antibiotics and your microbiome is destroyed, of course it will be followed with Candida albicans infection. Okay. Also in same in digestive tract, yeah. If we don't have normal microbiome of digestive tract, it might be followed with the microbiome with another microbiome which potentially dangerous for us. Okay, which examples of them? And some of them, in some situations, in immune deficiency, they can cause diseases. So in a normal site of action. So, for example, Staphylococcus aureus. So it's very widespread disease, widespread bacteria, but in some situations, the Staphylococcus aureus can cause a very severe cases of sepsis and septic shock later, and death, of course. We discussed it a little bit before. Then Streptococcus pyogenes. Streptococcus pyogenes, they can cause pyogenic papules all over the skin, so also can cause sepsis. Acne, so which is caused by Propionibacterium acnes. For example, tooth decay, so caused by Streptococcus mitus. So you see, this normal microbiome sometimes became start use us as a source of food. So that's that we have to prevent. Okay, so now, due to new techniques of bacterial identification relying on ribosomal RNA sequences have dramatically improved our understanding of the microbiome. In healthy individuals, the microbiome is very diverse. For example, there are estimated to be over 1,000 species of bacteria in the normal intestinal flora of an individual. So you see a lot of different species of bacteria, and they are like in political situations, they are always interacting with each other and counteracting with each other, and they struggle for their place in this area, and they mutually depend on each other. But if we kill at least one of this species, so one of species of this bacteria, so equilibrium is damaged, and then it finally causes a diarrhea, might cause diarrhea. It might be changed by diet and changed by environment and also by antibiotic treatment. That's why in case of antibiotic treatment, you have to resettle microbiome also. Anyway, every time you prescribe antibiotics, so please prescribe also probiotics to this patient, because when the normal microbiome is injured, So there are other bacteria can settle there, and it's dangerous for their life. Okay, the diversity of bacteria is greatest in the oral cavity and the stool, intermediate in the skin, and least in the vagina. So oral cavity, so why it's very diverse, so it's not it doesn't might create question, yeah. So because oral cavity is very closely related to outer environment. We take food, we talk, we yell, yeah, so we inhale air with the oral cavity. And in oral cavity settled a lot of different bacteria, and most successful of them, they survive there. That's why in oral cavity we have very widespread lymphatic capillaries, which prevent the bacteria, so very abundant immunity reactions, like reactions present, so immunity organs are present like tonsils, yeah, in oral cavity to prevent spreading of this bacteria. Okay, uh, so bacterial microbiomes at various body sites are partially similar in different individuals. Okay, um, let's talk about dysbiosis. So yeah, and dysbiosis is when normal flora is compromised with some other bacteria. So let's skip it. It's not so interesting. So which techniques we use for identifying of infectious agents? We can use bacterial culture. So we can take bacteria or fungi or any other, even sometimes viruses, and we can make a culture of them. So with on the feeding area of them, and then we can see how it grows, and we can see some features of it growth, and we can claim, so yeah, must be it is this type of bacteria, and we have to treat it in this way. Okay, histology. For example, for example, large bacteria like so which bacteria, for example? Yeah, so no, Escherichia coli can be found, but no, it's a better example. A good example is tuberculosis. Yeah, for example, tuberculosis. It can cause it can cause a specific reaction of our body. So you remember it's granulomatous inflammation with the formation of giant cells of Langhans giant cells, and this is would be the best example of histological examination. Also, and um, protozoas and other parasites, they can be well stained by hematoxylin and eosine. That's why in histological preparation we can easily see them. Uh, so in Candida and mucor among the fungi, so also can be visible. Okay, but for them we can use other stains like Giemsa stains, gram stains, acid fast stains in case of tuberculosis, silver stains, mucicarmine stains. Okay, so they can be used in histological examination, and we can see this their pathogenesis. Okay, in serology. So you know that reaction to the infection is made by our B lymphocytes. Yeah, so by formation of antibodies, and we can detect these antibodies in our body. So, and the fast reaction and in acute phase reaction, so we have, as we discussed, immunoglobulin EM secretion. Immunoglobulin M, so our plasma cells derived from B lymphocytes, they produce IgM first, and in the onset of disease. But later, this IgM is changed into the IgG, and by checking of by measuring all these antibodies, we can say, so in which phase of the disease is it right now. For example, if you can find only IgM, usually it means that disease just started. So that's why now in serologic tests of coronavirus, yeah, they always pay attention to IgM. So if IgM is very high, we can say that this is this coronavirus in progress yet. But if IgG is very high, but IgM is zero, so we can claim that a stable immunity is maintained there. Any question? So, sounds like someone wants to ask something. Imran, you have a question? No? Okay, let's continue. Then molecular diagnostic. Molecular diagnostic, by this molecular diagnosis is well known as the PCR. It's polymerase chain reaction. So very famous type of reaction. So by which we can reproduce the sequence of antibody, sequence of DNA or RNA. So let's it's all of this is based on principle of complementary principle. So for example, we can take DNA of the bacteria and compare it with DNA of with other bacteria. If they match in this in PCR test, so we can say, oh, it's PCR positive. So must be it matching. So it's matching due to complementary mechanism. So it matching. So must be is a similar DNA, and we can claim, yeah, this is chlamydia or some might be a mycoplasma. But if it doesn't match, we say, oh, it's a negative. So and here we have to have some probes of PCR, probes of PCR for every bacteria to check it specifically. And it's very specific test for this. And we can
Sir,
use smears, we can use blood, um, uh, we can use CSF even. Yeah, it's shown here. So that's idea.
correct. PCR expenses, sir.
Everywhere it's expensive. So because if country doesn't produce
Per
PCR
test,
testing
sir.
methods, so that's can be um, how it's called, it can be cheap, of course. Yeah. Okay, and by PCR we can usually we can find viruses which we couldn't find it in other with other methods. So, for example, intracellular parasites can be found, and also viruses like Epstein-Barr viruses. So they are assessed with the PCR CMV virus. So they're assessed with PCR. And also, when we check out the quantitative amount of viruses in blood, so we also use PCR method. For example, in hepatitis C, B, we check the PCR also to be sure that what's the level of viral load there. And then let's talk about proteomics. Proteomics, so it's very rarely used. So it's a mass spectrometry. It can be used to identify microorganism. We have in every bacteria, virus, or any kind of other cells which potentially injurious for us. They have some specific proteins, and if we identify these proteins, we can claim, yeah, this specific protein is present in this culture. So then must be it is this bacteria. So, but it's very rarely used in Kyrgyzstan. We don't have any kind of this check-in systems. But anyway, you have to keep it in mind that possibly we have proteomics, but because it's less effective than other. Um, uh, it's very useful in other countries might be, but in our country it's not useful because we have much more PCR methods and other methods. So but it's not useful for antibiotic sensitivity, so then it requires culture. That's why proteomics are not very important in diagnosis. Okay, so let's talk about a little bit so newly emerging and re-emerging infectious diseases. So first of all, I have to keep your attention in about Helicobacter pylori. So now, actually, this Helicobacter pylori makes a new challenge for us, a new way of treatment of gastritis and ulcer, so gastric ulcer approach. Because we used to thought, in old books you can find that gastritis and ulceration is caused by improper feeding, yes, so improper eating of food, so might be too spicy food, digestion, yeah. But along with it, so it's true, it's still true, but along with it, we found the causative agent of so along with this condition, so Helicobacter pylori, which is present in stomach, they also can cause they also can cause gastritis and ulceration of the stomach. It was found in the 80s, 1980s, and in 20, 2012, if I'm not mistaken, 2012. They got Nobel Prize because they correlate this Mycobacterium, Helicobacter pylori with the gastritis and mm, uh, gastritis and uh, ulceration, stomach ulceration. Okay, also we have Mycobacterium lepromatosis. So it's a new cause of leprosy. Leprosy is present, so it's a very rare condition, but still you have to know. It's a close relative of Mycobacterium lepra. Okay, it's not well. It's very rarely present. Okay, so here you see. So it's before coronavirus infection in its book of 2018. So, but it talks about Middle East respiratory syndrome coronavirus. It's MERS-CoV. MERS-CoV, MERS-CoV 1. Yeah, so now we encounter with MERS-CoV 2, but now it's a MERS-CoV 1, Middle East respiratory syndrome, and SARS. It's called severe acute respiratory syndrome, like SARS-CoV. Yeah, likely spread to humans from animals, and were first detection in 2003 and 2012 respectively. Okay, let's continue. Microorganisms can acquire genes that enhance virulence or overcome host defense. Example, 4,000 individuals in Germany were infected. Yeah, 2011, that was a widespread of Escherichia coli that spread in sprout seeds. New strain was derived from a different type of Escherichia coli that acquired a gene for the Shiga toxin from a bacteriophage. So, okay. And other pathogens have become much more common because of immune suppression caused by AIDS. So previously AIDS was not present, yeah, and that our immunity usually fight against this viruses or bacteria or fungi. But now in these patients, these bacteria and viruses, they can spread much faster and cause specific infections, and now amount of these infections are much larger. Okay, here they talk about Zika virus in Uganda. So might be okay, AIDS, Zika virus against agents of bioterrorism. So which of them we can discuss? Which bioterrorism is used? We have we have A, B, and C designated. Okay, but now it's banned. I hope no one will ever use this bioterrorism. So it's very hard to produce it in by ourselves. So it's used to be influence of government to production of this bacteria, which can be used as a bio weapon, biological weapons. So that's why I hope we can leave it. Okay, and in next lecture, tomorrow lecture, we'll start with the transmission and dissemination of microbes, and how dissemination happens and transmission in these cases. Okay, for today it's enough. So let's check your attendance. Okay, I checked. So now let's check your names in the chat. Okay, that's it. Thank you for your attention. See you next time. Goodbye.